Imagine a world where disease and sickness are detected and diagnosed instantaneously. One in which where past and early detection are but a mere formality. That future isn't too far off with the help of nanobiosensors. Let me explain. From year to year, cancer and other diseases become untreatable due to the delay in diagnosing and detecting them correctly. In the year 2014 alone, 855,000 new cases of cancer were discovered in the United States. 855,000 is quite a lofty number, so the importance of early detection cannot be overstated. In addition to this pivotal contribution, biosensors hold immense power in our daily lives, limitless possibilities. Nanosensors are chemical or mechanical sensors that can be used to detect the presence of chemical and biological species, as well as physical parameters, like temperature, but on the nanoscale.
Bio-nanosensors specifically detect protein, antibodies, and other biologically relevant molecules. Bio-nanosensors can mimic the complexity of biological systems currently operating in you and I. These systems, however, are very complicated. For example, there are over 86 billion neurons in the average human male brain. The applications of bio-nanosensors can range from drug screening and discovery, point-of-care modification, and even diagnostics. They're really limitless. The abilities of bio-nanosensors are endless. Disease detection, food allergy monitoring, environmental health screening, and more. Who knows where bio-nanosensors could take us?
Wow, could you imagine that kind of future? The real question for me is, how is it done? How can you achieve order and precision at such small scales? How do we connect biological molecules to nanoscale circuits while preserving function and not compromising structure? So how do we ensure the fidelity of the information transfer between the parts of a bioelectronic circuit? And lastly, how do we create an interface between that circuit and a complex organism like a cell?
This perfect harmony between bio and nanoscale electronics has the potential to process extremely complex functions, like detecting disease markers at an impossibly small concentration, or even reconstructing the biological function of damaged neurons on a chip, or extend beyond into extrasensory functions. In order to understand how these nanosensors are made, we must take a look inside the toolkit used by researchers in the field of nanobioelectronics.
Sensors consist of the same general structure, which include the detection of a biosample, a recognition element to detect that sample, a transducer to change the signal into processable information, and a signal processor to display that data. Researchers in this field have no lack of resources, as they're able to use materials such as nanowires, thin films, and chip-based architectures. They get to pick and choose the best of both worlds, with each offering its unique advantages and limitations. All right, so we have access to biomolecules and advanced inorganic materials. But what now? Modern developments have recently focused on field electric transistors, FETs. They implement an electric field to control the flow of electrons from source to sink. All FETs have a source, drain, and gate terminals. Electrons flow from the source through the gate to the drain. In the case of nanotechnology like nanowires, FETs imitate microarrays. Here is a physical layout of an FET.
A carbon nanotube field effect transistor refers to an FET that utilizes a single carbon nanotube or an array of carbon nanotubes as the gate material. Research has shown that the nanotube and nanowire transistors have proven to be the most suitable for biosensing applications.
Silicon nanowires, with their friendly, biocompatible silicon oxide surface, have emerged as leaders for applications in bio-nanoelectronics. These silicon nanowire transistors can operate in ionic solutions, which is native environment for most biological systems. They can provide a natural and effective interface between the microscopic device and macroscopic equipment. Also, one silicon chip can house thousands of devices, providing the ability to run parallel measurements. These chips can be placed on flexible polymer substrates, further enabling interfacing with complex tissues. Lastly, and most importantly, the transistor's gain provides an effective means of amplifying the weak signal from low concentrations of biomaterials, allowing for detection of potential disease markers. So you've assembled your miniature transistors using silicon nanowires and are ready to start detecting. But how do you actually detect your target molecules or events? In this video, we'll go over a few applications of this field along with some limitations and future prospects.
Currently, if I wanted to record electrical impulses on my heart cells, I would introduce the traditional patch clamp micropipette to myself. The patch clamp technique implements a micropipette which clamps onto a cell and can measure voltage across the membrane, but the patch clamp is limited by tip size and has poor spatial recognition. Although effective in recording nerve pulses, microelectronic arrays, which are metallic or inorganic, are rigid. Because of the complex structure of the heart, microelectronic arrays may abrade surrounding cells due to their rigid shapes and can reposition over time. This problem has pushed research into more flexible transducers.
Recent developments have centered around nanoelectronics integrated with flexible polymer substrates.
One specifically is nanowire FETs. Nanowire FETs forego the planar geometry of microelectronic arrays for a more linear one. What this entails is the option to create arrays onto a flexible polymer substrate. Nanowire FETs demonstrate better cell-device interaction and have higher spatial resolution because of their flexible nature. Although nanowire FETs are able to conform to our organ's shape because there is no direct probing of cells, the traditional patch clamp method is able to record heartbeats of an average 100 milliseconds faster according to Dr. Timiko's measurements.
The common type of bioelectronic sensor on the nanoscale is the carbon nanotube and silicon nanowire. Attaching various detection methods onto the nanowires allows us to detect binding. For instance, researchers interested in early cancer detection can attach cancer-specific antibodies to carbon nanotubes.
These specific antibodies can be immobilized and anchored on the carbon nanotubes with two different approaches. One, direct non-covalent bonding of antibodies to the devices, or two, bonding the antibodies covalently or non-covalently to the polymer previously attached to the carbon nanotubes, FETs. After introducing the solutions of interest, researchers can measure whether binding events occur. If no binding events occur, then the solution is free of target antigen. If many binding events occur, then the solution has the antigen. In some research, researchers has created arrays of carbon nanotubes to measure a variety of things at once. You can see how this method has expanded to various diseases or even industrial applications. Electrochemical monitoring of biorecognition events provides a promising platform which is simple, cost-effective, and requires no external modification on the biomolecules. In one study, carbon nanotubes were modified with aptamers and monoclonal antibodies in order to detect immunoglobulin E at 250 picomolar quantities. The aptamers used consist of synthetic oligonucleotides that were generated to recognize immunoglobulin E, but they can be modified to recognize specific amino acids, drugs, and other proteins of high specificity. Immunoglobulin E is capable of triggering powerful immune reactions, thus its rapid detection is of great interest in treating patients with allergy-mediated disorders. Other studies have also focused on the detection of disease-causing agents such as Salmonella and E. coli.
A limitation of these approaches include uneven distributions of targets within the solution. This can cause recording of false signals, especially when only small amounts of our signal molecule are in contact with the FET. Large number of samples can be taken. However, in order to average these results and increase accuracy, you would have to increase the size of the device, and this limits the nanoscale properties.
Also important is whether or not the target is placed within the die-by length of the FET. An FET's die-by length is simply the distance required for screening the surplus charge by the mobile carriers present. Thus, for the surplus charge to be effectively measured, a target must be within the die-by length of the FET. With this in mind, the aptamer used in the FET must be below its die-by length, which unfortunately limits its size, affecting which targets can be identified. In another example, to detect performance-enhancing drugs, stanzolol and methylboldenone were immobilized on the carbon nanotube structures in FET arrays for anabolic androgenic steroids. To combat electronic interference, polymer modifications were proposed to minimize the aggregation of nonspecific adsorption onto carbon nanotubes. Without reducing nonspecific adsorption, the results can lead to false positive responses in the transistors. Thus, its applications are limited to purified samples.
Researchers have developed multiplex electrical detection of cancer using silver nanowire FETs with wires and surface receptors. Arrays of silver nanowires were created on a substrate and monoclonal antibodies were attached to the wires.
A change in conductance signals a binding event. In human and donkey serum, prostate-specific antigen was detected at femtomolar concentrations. Previous techniques, such as ELISA, surface plasmon resonance, nanoparticles, microcantilevers, and carbon nanotubes, have not achieved such specificity. The data shows that direct detection of prostate-specific antigen is routinely achieved for concentrations under 2 femtomolar. Additionally, telomerase has recently become a cancer marker of interest. Telomerase activity has been found in at least 80% of all known human cancers, but no activity is found in the adjacent somatic tissues. Thus, telomerase has been proposed as a marker for cancer detection and as a therapeutic target. By using this method, researchers can detect telomerase quickly and accurately.
Our future may render hospitals obsolete, a time when Bionanoelectronics are so cheap and affordable that any one person can walk to a local convenience store and buy some apparatus and diagnose/test themselves at home. But it goes beyond the human body. It reaches into the deepest roots of our environment. Real-time assessment of the air we breathe, the soil which we grow crops, or the water we drink. This would be a time where food would be in abundance, suns would be extinct, and water would no longer be poisoned, and the list goes on and on. In essence, bionanoelectronics would result in a real-life Elysium. We think something as big as utopia could become modern day as a result of something as small as bionanoelectronics.